Hello everyone, welcome back to the channel. In today's lecture, we will be covering the physical method of sterilization, including heat sterilization like moist and dry heat, filtration, radiation, and mechanical methods. We will also discuss their principles, mechanisms, applications, and some important points you need to know. So let's get started. Sterilization simply means killing or removing every single living organisms or microorganisms from an object, substance, or environment, including tough bacterial spores that usually resist normal cleaning. It's absolutely critical in healthcare, labs, and even industries because contaminated instruments or solutions can lead to serious infections or loss of product quality. There are two main categories of sterilization methods: physical methods and chemical methods. Today, we focus on physical methods using heat, filtration, radiation, and mechanical forces. Physical methods can be divided into four major categories. First, heat sterilization, which is most reliable and widely used. Second, filtration, for when heat would destroy the material, like vaccines. Third, radiation, which can sterilize even packaged material. And fourth, mechanical removal methods like washing and filtration systems. Let's start with heat, the most common method. Now let's take a detailed look at heat sterilization, one of the oldest and most trusted methods of sterilization. First, let's understand the principle behind it. Heat sterilization works by applying high temperatures that cause irreversible damage to the microorganisms. Here is how. It causes denaturation and coagulation of proteins, meaning the critical enzymes and structural proteins inside the cell lose their natural shape and clump together, making them non functional. It also disrupts the integrity of cell membranes and cell walls, causing leakage of cell contents and loss the essential cellular function. Additionally, heat can cause oxidative damage, burning or oxidizing important molecules inside the cell. In the cause in case of dry heat, there is also dehydration, the removal of water from cells, leading to shriveling and death. So the final result is the complete destruction or inactivation of all forms of microbial life, including those highly resistant bacterial spores. Now, when we talk about heat sterilization, it's mainly divided into two types. Moist heat sterilization, where we use water vapor like steam or boiling water to carry and transfer heat efficiently. Moist heat is more effective because the presence of water allows for better penetration and faster protein denaturation even at lower temperature. Dry heat sterilization, where we use hot dry air. This method needs higher temperatures and longer exposure times because air is less efficient than water at transferring heat. Both methods are incredibly useful depending on the material being sterilized and the required level of sterilization. Now, in the next sections, we will dive deeper into each method separately, starting with moist heat sterilization. All right, let's now dive into moist heat sterilization. Moist heat sterilization simply means using hot water or steam to kill microorganisms. Unlike dry heat, moist heat transfers heat more efficiently because water conducts heat far better than air. This allows us to achieve sterilization at lower temperature and in shorter times. The main way moist heat works is by coagulating and denaturing the proteins inside the microorganisms. Remember, proteins are like the machinery inside every living cell. When heat messes up their structure, the cells can no longer survive. Because of its efficiency and reliability, moist heat is widely used for sterilizing culture media, surgical tools, hospital linen, pharmaceutical solutions, and many more items. Now let's understand the mechanism how exactly moist heat kills the microbes. When steam comes into the contact with a cooler surface, like a surgical instrument or a culture flux, it condenses into water. During this condensation, it releases a huge amount of latent heat, more than just the heat of regular steam. This sudden release allows rapid and deep penetration of heat into the material. This intense heat leads to denaturation of protein, inactivation of critical enzymes, and disruption of cell membrane. Ultimately, this cascade of damage causes complete microbial death. Even highly resistant bacterial spores can be destroyed if we maintain high enough temperatures for the right amount of time, like in an autoclave. Now, moist heat sterilization is applied through several different methods, including pasteurization, boiling, Tyndallization, which is also called intermittent sterilization, and autoclaving, which is sterilization under pressure. There are different moist heat techniques based on the temperature. Below 100 degree Celsius, we mainly do pasteurization for foods and inspection for delicate media. At 100 degrees Celsius, boiling and Tyndallization are used. Tyndallization is special because it allows spores to germinate and then kills them. Above 100 degrees Celsius, using pressurized steam, we have the autoclave that utilizes the sterilizer. Now let's look into some important moist heat methods, namely pasteurization, boiling, and Tyndallization. First, we have pasteurization. It's a method of applying mild heat, usually below 100 degrees Celsius, just enough to kill harmful pathogens, especially those that cause disease, but without destroying the quality of the product. Pasteurization is mainly used for milk, juices, beer, and wine. There are two main methods: the holder method, where the product is heated at 63 degrees Celsius for 30 minutes, and the flash method, which is faster, heating at 72 degrees Celsius for just 15 seconds. Note that pasteurization is not complete sterilization. Some harmless bacteria may survive, but the microbial load is greatly reduced to safe level. Next, we have boiling. Boiling water at 100 degrees Celsius for 10 to 30 minutes can kill most vegetative bacteria, fungi, and many viruses. However, it does not reliably kill all the bacterial spores, which are highly heat resistant. Thus, boiling is more suitable for disinfection rather than guaranteed sterilization, useful for drinking water, basic instruments, and fabrics. Finally, there is Tyndallization or intermittent sterilization. Here we heat the material at 100 degrees Celsius for about 20 minutes but repeat the process over 3 successive days. Why 3 days? Because spores that survive the fast heating get time to germinate into vegetative cells overnight, and these vegetative forms are easily killed during the next heating. Tyndallization is specially useful for heat sensitive materials like sugar media, gelatin media, or some plant extracts, where you cannot use harsher methods like autoclaving. Now let's discuss the king of moist heat sterilization, autoclaving. So autoclaving uses steam under pressure to achieve very high temperatures, much higher than what boiling water can reach at normal atmospheric pressure. Remember, at standard atmospheric pressure, water boils at 100 degrees Celsius. But if we increase the pressure, water can stay liquid and reach higher temperatures like 121 degrees Celsius without boiling away. This combination of high heat and high moisture is extremely lethal to microorganisms, including resistant bacterial spores. The standard operating conditions for an autoclave are 121 degrees Celsius, 15 pounds per square inch pressure, and 15 to 20 minutes exposure time. Of course, the exposure time depends on the load size and the type of the material you are using. There are different types of autoclaves: gravity displacement autoclaves, where steam pushes the air out by gravity; pre-vacuum autoclaves, which uses a vacuum pump to actively suck out all the air before introducing steam. This improves steam penetration and speeds up the sterilization process. Autoclaves are widely used for sterilizing surgical instruments, culture media, glassware, rubber goods, and even certain plastics. Some important precautions to keep in mind: always ensure air is completely removed from the chamber, otherwise, air pockets can prevent proper sterilization. Don't overload the autoclave. Steam needs space to circulate freely. And finally, verify that sterilization has been achieved, typically using biological indicators like Bacillus stereothermophilus spores, which are highly resistant and a good test for proper sterilization. Autoclaving is considered the gold standard for sterilization, reliable, fast, and safe when properly operated. Dry heat sterilization means using hot, dry air to kill microorganisms. Unlike moist heat, dry heat lacks water or steam, and hence the transfer of heat energy is slower. Because of this, we need to use higher temperatures and longer exposure times to achieve the effective sterilization process. Dry heat is ideal for materials that would be damaged by moisture, such as powders, oily substances, glassware, and metal instruments. Now, how does dry heat work? The mechanism. At high temperature, dry heat causes oxidative destruction of essential cellular components. In simple terms, it burns or oxidizes vital parts of the microbial cells. It also causes dehydration, removing water from cells, leading to structural collapse. Finally, it denatures proteins by removing the critical water molecules necessary to maintain their structure. However, microbial death by dry heat is usually slower compared to moist heat sterilization. That's why we use higher temperatures for longer period of time. Now let's see the common methods of dry heat sterilization. Now let's focus on one of the most commonly used equipment in dry heat sterilization is hot air oven. The principle behind the hot air oven is simple. It uses dry heat at high temperatures over a sufficiently long time to achieve sterilization. The killing of microbes occurs by protein denaturation, oxidative destruction of vital cell components, and dehydration. The standard sterilizing conditions inside a hot air oven are 160 degrees Celsius for 2 hours, 170 degrees Celsius for 1 hour, 180 degrees Celsius for 30 minutes, depending on the material and the need. It is particularly suitable for sterilizing glassware like test tubes, flasks, petri dishes, metallic surgical instruments like scissors and scalpels, heat stable powders like starch or talcum powder, and fats and oils that cannot be sterilized by moist heat. Some important points while using the hot air oven: make sure all items are completely dry before loading. Moisture can interfere with proper sterilization. Proper air circulation is vital. It ensures even heating throughout the chamber. After sterilization, allow the oven and materials to cool down gradually to avoid sudden thermal shock, which can crack glassware. Moving on to two simple but very powerful sterilization methods: flaming and incineration. So, flaming is the quick sterilization technique where we expose an object directly to an open flame. The intense heat instantly kills any microorganisms present on the surface. This is routinely used in microbiology labs for sterilizing inoculating loops and needles and flaming the mouths of culture tubes or flasks before and after the transferring cultures. It's fast, easy, and very effective when handling small items. Next, we have incineration, which is the complete burning of contaminated materials. Incineration is carried out at extremely high temperatures, sometimes up to 870 to 980 degrees Celsius, completely oxidizing and destroying microorganisms, toxins, and even the material itself. This method is widely used for medical waste disposal, such as infected dressings, pathological wastes, animal carcasses, and destruction of disposable contaminated materials like syringe, cotton, and gloves. There are specialized medical incinerators and laboratory incinerators are designed to handle different kind of biological wastes. Filtration works on simple but powerful principle. It physically removes microorganisms from liquids or air by passing them through a filter with very tiny pore size. There are different types of filter depending on what we are trying to sterilize. Depth filters made of thick fibrous materials, they trap microorganisms within their tangled structure. These are good for heavy particle load. Membrane filters, thin and made of cellulose esters or other polymers. These have precisely defined pore size, usually 0.22 micron or 0.45 microns, ideal for sterilizing liquids such as antibiotics, vaccine, and culture medias. HEPA filters, high efficiency particulate air filters, which are used for sterilization of air or to sterilize air. They remove 99 to 99.97% of particles that are zero point 0.3 microns or larger. HEPA filters are installed in operating rooms, biosafety cabinets, and clean rooms. Some important points. Remember that filtration does not kill microorganisms, it just removes them from the fluid or air. Regular checking and validation of filters is essential to ensure the sterility. Now let's dive into radiation sterilization, a method where we use energy waves to destroy the microorganisms. The basic principle is that radiation damages the DNA and other essential components of microbes, preventing them from reproducing and leading to their death. There are two main types of radiations. Ionizing radiation. This includes gamma rays, X-rays, electron beams. These are high energy waves that cause ionization, meaning they break chemical bonds inside the microbial DNA and proteins, leading to irreversible damage. Ionizing radiation is highly penetrating, meaning it can pass through materials and sterilize even deep inside. Applications include sterilization of disposable medical supplies like syringes, catheters, surgical gloves, pharmaceutical sterilization, and also food preservation by eliminating pathogens and increasing the shelf life. Non-ionizing radiation. This mainly refers to ultraviolet light. UV light is lower energy and works by um, creating thymine dimers in microbial DNA, preventing replication. However, UV has poor penetration. It can only sterilize surfaces, air, and water. Wherever direct light exposure is possible. So, some important points to remember that ionizing radiation is widely used industrially. UV radiation needs direct line of sight to be effective. Any shadowed area may remain unsterilized. Now let's talk about mechanical methods of sterilization, where we use physical forces to remove microorganisms from the surfaces, liquids, or air. The principle here is simple. We don't actually kill the microorganisms, we physically remove or separate them. Let's look at some points or important points of mechanical methods like filtration. We have already discussed this. Microbes are removed by passing fluids or air through fine filters. Scrubbing and cleaning. This involves manually removing dirt and microbes using detergents, brushes, and waters. For example, scrubbing surgical instrument before autoclaving them. Sedimentation. Here we allow microorganisms and particles to settle naturally under the influence of gravity. And then we remove the clear upper portion. Applications include pre-cleaning surgical instruments before sterilization, air purification process, water treatment plants, preparing sterile pharmaceutical solutions. Key point to remember is that mechanical methods are not killing microorganisms. They just reduce the number and prepare materials for more effective sterilization later. All right, let's quickly summarize everything we have discussed today. Sterilization is the process of completely eliminating all forms of microbial life, and it is a cornerstone for ensuring safety in healthcare, research, food, and pharmaceutical industries. Among the physical methods of sterilization, we learn heat methods, but both moist heat like autoclaving and dry heat like hot air ovens and incineration, filtration for removing microbes from air and liquids, radiation such as gamma rays and UV light for deeper and surface sterilization, mechanical methods like scrubbing, cleaning, and filtration to reduce microbial load. Key points to remember: most heat sterilizes quickly and efficient. Moist heat sterilizes quickly and efficiently. Dry heat is perfect for items that cannot tolerate moisture. Filtration is the go-to for sterilizing delicate fluids and air. Radiation provides industrial level of sterilization solutions. Mechanical methods help prepare instrument and surfaces for better sterilization outcome. So the choice of sterilization method always depends on the type of material, the type of microorganisms, and the specific purpose you are aiming for. Thank you so much for watching this lecture. If you found this helpful, make sure to give it a like, share it with your friends, and most importantly, subscribe to the channel for more detailed and easy to understand microbiology lectures.